This poster presentation is called Investigating the Role of the Gut Microbiome as a Mediator for Radiation Therapy-Induced Bowel Injury, and my name is Alex Jameus. Before beginning my poster, I would just like to add that I have nothing to disclose, no affiliations with any for-profit or not-for-profit organizations, and the financial support for this project has been received through NSERC, CAHR, and NHRC funding agencies. The goal of this research project is to observe how radiation affects the gut microbiome and gastrointestinal tract itself. Over half of all cancer patients will undergo radiation therapy, which is associated with a long list of adverse effects that largely affects patient quality of life. The low tolerance of abdominal and pelvic organs, along with the high doses of radiation needed to abolish a tumor in this area, leads to permanent changes in bowel habits in as many as 90% of patients following treatment. We believe that an overlooked component of radiation-induced bowel injury is the role of the gut microbiome in tissue health and repair. To analyze the effects of radiation on the gut, the gastrointestinal tract of male mice was collected 48 hours following various doses of ionizing radiation exposure. Overall, radiation exposure did not lead to significant changes in sample diversity or bacterial phylum composition 48 hours post-irradiation. However, the ileum shows early signs of GI toxicity at the high-dose treatment we used. We are currently optimizing a system which will allow for the genetic engineering of Lactobacillus reuteri, an endogenous species of the gut microbiome, to deliver factors that can aid in the recovery of gastrointestinal tissue following radiation exposure. To do this, a gene of interest and an antibiotic resistance will be put into a parent plasmid to make unique clones. This has already been completed for the first of the clones and verified through both colony PCR and functional verification through the imaging of a green fluorescent protein. Eventually, the gastrointestinal tract of mice will be inoculated with strains of our genetically engineered bacteria, followed by having targeted radiation treatments to the gastrointestinal tract and assessed to reveal if there has been success in mediating radiation-induced bowel injury.